There are communities in the modern world that function, for the imagination of the people living outside them, less as actual human societies with the full complexity and contradiction that phrase implies, and more as symbols, living emblems of an idea or an argument or a way of answering a question that the surrounding culture cannot stop asking about itself. The Amish have occupied this symbolic position in the American imagination for so long and so completely that the actual human beings living in the settlements of Lancaster County and Holmes County and the dozens of other Amish communities distributed across the eastern and midwestern United States have become almost invisible behind the image that has been constructed around them. The image is familiar to the point of cliché: plain clothing, horse-drawn buggies, hand-built barns, oil lamps, and a principled rejection of the technological infrastructure that defines modern life. A community that stepped off the conveyor belt of historical progress at some point in the 18th or 19th century and has been standing quietly to the side ever since, watching the rest of the world accelerate past them with an equanimity that strikes outside observers alternately as admirable, puzzling, and faintly reproachful. What that image conceals, and what modern genetic research has now begun to reveal with a clarity and a specificity that no amount of historical or sociological investigation had previously managed to provide, is that the Amish are not who the popular imagination has assumed them to be, not in their biological ancestry, not in the trajectory of their demographic history, not in the genetic legacy they carry, and not in the medically significant biological characteristics that their centuries of extraordinary insularity have concentrated in their population in ways that are simultaneously scientifically extraordinary and practically urgent. The DNA tells a story about the Amish that is more complicated, more historically layered, and more consequential than the simple narrative of Swiss and German religious refugees, preserving a static tradition in the New World that has dominated both popular and scholarly understanding of the community for generations. And the implications of what that story reveals reach far beyond the boundaries of Amish settlements into fundamental questions about human genetics, population history, disease biology, and the relationship between cultural practice and biological destiny that medical science is only now developing the tools to fully address. To understand what the genetic research is revealing, and why it matters as much as it does, you need to understand what the Amish actually are in historical terms. Not the symbol, but the community, with its specific origins, its specific demographic trajectory, and the specific set of circumstances that have made it one of the most genetically distinctive human populations on the surface of the North American continent. The Amish trace their origins to the Anabaptist movement that emerged in the Swiss and South German Reformation of the early 16th century, a radical wing of the Protestant Reformation that rejected infant baptism, insisted on the separation of church and state, refused to swear oaths or bear arms, and demanded a level of communal discipline and separation from the surrounding world that brought its adherents into immediate and violent conflict with both Catholic and Protestant civil authorities throughout the 16th and 17th centuries. The persecution that the early Anabaptists experienced was not mild or theoretical. It was systematic, sustained, and lethal, involving torture, drowning, burning, and beheading carried out by civil authorities across the Swiss Confederation, the German principalities, and the Habsburg territories, with a thoroughness that drove the surviving communities into increasingly remote and marginal territories where they could maintain their distinctive religious practice at the price of extreme geographic isolation. The specific Amish identity, as distinct from the broader Anabaptist and Mennonite tradition, crystallized in the 1690s around the figure of Jacob Amann, a Swiss bishop whose insistence on stricter discipline and more complete separation from the world produced a schism with the more moderate Mennonite communities that has never healed. The followers of Amann, the Amish, maintained the most rigorous version of the Anabaptist commitment to separation, developing the distinctive set of practices and prohibitions known as the Ordnung that governs every aspect of Amish life, and that has been the mechanism by which Amish identity has been transmitted and enforced across every subsequent generation. When William Penn's experiment in religious toleration in Pennsylvania began attracting European religious refugees in the early 18th century, Amish and Mennonite families were among the earliest to emigrate, and the first Amish settlers arrived in Pennsylvania beginning around 1737, concentrating initially in the Lancaster County area, where the largest and most famous Amish community in the world still exists today. What happened to the Amish population after those initial waves of 18th century immigration is the demographic foundation of everything that modern genetic research has been uncovering, and it is a story of population dynamics so unusual, so extreme, and so consequential in its biological effects that it has no close parallel in any other surviving community in the contemporary United States. The Amish do not seek converts. They do not proselytize. Membership in the community is acquired by birth and retained by the individual choice, made in young adulthood during the period of exploration known as Rumspringa, to accept baptism into the Amish church and commit to life within the Ordnung. Those who choose not to return, and the proportion who leave varies significantly by community and by generation, exit the gene pool of the community permanently, because the social mechanisms of shunning that the Ordnung prescribes for those who leave after baptism effectively prevent the kind of partial reintegration that might otherwise allow genetic material to flow back into the community from the outside world. The result is a population that has been reproductively isolated to an extraordinary degree for approximately 300 years, drawing exclusively on the genetic material present in the small founding population that arrived in the 18th century with essentially no genetic input from the surrounding population of non-Amish Americans across that entire period. The genetic consequences of this prolonged and extreme isolation are what the modern research has been characterizing in increasing detail, and they begin with a concept that population geneticists call the founder effect, the mathematical reality that when a large population is established from a small founding group, the genetic diversity of the resulting population is constrained by the genetic diversity that happened to be present in that founding group, regardless of how large the subsequent population becomes. The Amish founding population in 18th century Pennsylvania was small. Estimates based on historical records and genetic analysis suggest that the entire founding group from which the contemporary Amish population descends numbered somewhere between 200 and 500 individuals, and that the effective population size, the number of individuals who actually contributed meaningfully to the genetic inheritance of subsequent generations, was smaller still, perhaps as few as 50 to 100 people, whose lineages became the dominant genetic threads in the community's subsequent history. 300 years of exponential population growth from that tiny founding base, combined with the near total absence of genetic input from outside the community, has produced a population that today numbers somewhere between 350,000 and 400,000 individuals, a number that has been doubling approximately every 20 years due to the Amish's extraordinarily high birth rate and their remarkable retention of community members despite the availability of the outside world during Rumspringa, but that carries the genetic diversity of a population a fraction of that size. Every Amish person alive today is, in genetic terms, closely related to every other Amish person, to a degree that would be considered remarkable in any non-isolated population, but that is the simple mathematical consequence of 3 centuries of descent from a few hundred common ancestors. What the DNA analysis has revealed about those founding ancestors is where the story becomes genuinely surprising in ways that challenge the conventional understanding of Amish origins. The popular narrative of Amish ancestry is straightforward. Swiss and South German Anabaptists, predominantly of Germanic ethnic background, arriving in Pennsylvania in the early 18th century and establishing the genetic foundation of all subsequent Amish generations. This narrative is not wrong, exactly. It correctly identifies the geographic and religious origins of the founding community, but it is considerably less complete than it appears. And the incompleteness matters because it conceals a genetic heritage that is both more diverse in its origins and more complicated in its implications than the simple Swiss-German story suggests. Genomic analysis of Amish populations, conducted by research teams at institutions including Johns Hopkins University, the University of Maryland, and the Clinic for Special Children in Strasburg, Pennsylvania, an institution that has been providing medical care to Amish and Mennonite communities for decades, and that has accumulated one of the most comprehensive databases of Amish genetic information in existence, has revealed that the Amish founding population carried genetic lineages that do not trace exclusively or even predominantly to the Swiss and South German populations that the historical narrative emphasizes. The admixture analyses show significant genetic components from populations across a much broader range of the European continent, including components associated with French, Dutch, and Alsatian populations. The last of these particularly interesting, given that Alsace, the borderland region between France and Germany that has changed national identity multiple times in its history, was a major staging ground for Anabaptist communities fleeing persecution from both directions, and represents a genetic mixing zone where Swiss, German, and French lineages were thoroughly combined in the 17th century refugee communities from which many Amish founders descended. More striking still is the evidence that the Amish founding population included genetic lineages with origins considerably further east than the Swiss-German heartland of Anabaptist origins. Lineages consistent with Central European and Eastern European populations that entered the Anabaptist community through the complex patterns of conversion, refugee movement, and community absorption that characterized the Anabaptist diaspora during the century and a half of persecution that preceded the Pennsylvania emigration. The Anabaptist communities that survived the 16th and 17th century persecutions did so by moving repeatedly, forming new communities in marginal territories, absorbing refugees from other persecuted groups, and intermarrying across what had originally been distinct ethnic and linguistic communities in ways that created a genetic heritage considerably more diverse than the simple label of Swiss-German religious refugees implies. The medical implications of the genetic story that the DNA is telling about the Amish are where the research has its most immediate and most urgent practical significance, and they have been the primary driver of the serious scientific attention that Amish genetics has attracted over the past 3 decades. The combination of a small founding population, centuries of isolation, near zero genetic input from outside the community, and a very high birth rate has produced in the Amish population exactly the conditions that population genetics predicts will lead to the accumulation of rare genetic variants, both harmful and, in some remarkable documented cases, beneficial, at frequencies dramatically higher than those observed in the general population. The same founder effect and genetic drift that have made the Amish genetically distinctive have also made them a uniquely valuable population for the study of gene function, because rare variants that would be nearly impossible to study in the general population, where they might affect one person in 10,000 or one in 100,000, are present in the Amish at frequencies high enough to allow rigorous investigation of their biological effects. The catalog of genetic conditions that occur at elevated frequencies in Amish populations is extensive and has been documented in detail by researchers who have worked with these communities, most notably D. Holmes Morton, the founder of the Clinic for Special Children, whose decades of medical work with Amish families gave him both the clinical data and the community relationships necessary to conduct genetic research of a kind that would have been impossible for outside researchers working without that foundation of trust. The conditions include glutaric aciduria type 1, a metabolic disorder that causes progressive neurological damage if untreated, but that responds well to early intervention. Maple syrup urine disease, another metabolic disorder with devastating neurological consequences that is treatable when identified before symptom onset. Ellis Van Creveld syndrome, a skeletal dysplasia that causes short stature and polydactyly, and that occurs in the Lancaster Amish at a frequency roughly 200 times higher than in the general population. And a range of other recessive conditions that are rare in the general population, but common enough in specific Amish communities to represent a genuine public health challenge that the Clinic for Special Children was specifically created to address. But the genetic story of the Amish is not exclusively or even predominantly a story of disease burden, and the research that has emerged over the past decade has increasingly highlighted an aspect of Amish genetics that is as biologically extraordinary as the elevated disease frequencies, but that has received considerably less popular attention because it does not fit the narrative of genetic isolation as straightforwardly harmful. In 2017, a research team led by Sadiya Khan and Simon Meyerson at Northwestern University published findings from a study of the Old Order Amish community in Bern, Indiana, that stopped the cardiovascular genetics research community cold with the specificity and the magnitude of the effect they had documented. The researchers had identified a mutation in the SERPINE1 gene, the gene that encodes plasminogen activator inhibitor 1, or PAI1, a protein that plays a central role in the regulation of blood clot formation and dissolution, that was present in heterozygous form in roughly 1 in 8 members of the Bernie Amish community, an extraordinarily high carrier frequency for a mutation that is essentially absent from the general population. The biological effect of carrying one copy of this mutation, heterozygous carriers, the vast majority of the mutation's carriers in the Bern community, was, in the data the research team collected, dramatic and consistent in a direction that was the opposite of what disease genetics usually documents. Heterozygous carriers of the SERPINE1 mutation had PAI1 levels approximately half those of non-carriers, and they showed a constellation of physiological characteristics that read, in the language of cardiovascular medicine, as a profile of substantially reduced aging-related deterioration. Their telomeres, the protective caps on chromosomes that shorten with age and whose length is used as a marker of biological aging, were measurably longer than those of non-carriers of the same chronological age. Their fasting insulin levels were lower, their metabolic function was better preserved, and their incidence of diabetes was significantly reduced. Most strikingly, the carriers appeared to live longer than non-carriers, by a margin that the researchers described as significant, and that has prompted a wave of investigation into PAI1 as a potential target for aging-related pharmaceutical intervention. The SERPINE1 finding illustrates something about the genetics of isolated populations that the disease-focused literature has historically underemphasized. The same founder effect and genetic drift that elevate the frequency of harmful recessive mutations in isolated populations also elevate the frequency of rare beneficial variants, and the biological consequences of those beneficial variants can be as dramatic and as medically informative as the disease consequences of the harmful ones. The Amish, in other words, are not simply a population in which genetic isolation has accumulated a burden of rare diseases that requires medical management. They are a population in which genetic isolation has created a natural laboratory for the study of human biology, in which rare variants affecting clotting, metabolism, aging, disease susceptibility, and a range of other physiological systems are present at frequencies high enough to study rigorously, and in which the cultural stability of the community provides the kind of longitudinal research opportunities that are simply unavailable in more mobile and more genetically heterogeneous populations. The picture of the Amish that emerges from the totality of modern genetic research is therefore considerably more complex, more historically layered, and more scientifically significant than the popular image of a static, isolated community preserved in amber since the 18th century. The founding population carried a genetic heritage that was itself the product of a century and a half of persecution, migration, intermarriage across ethnic boundaries, and community formation under conditions of extreme stress. A heritage more diverse in its European origins and more complicated in its biological contents than the simple Swiss-German narrative suggests. The 3 centuries of subsequent isolation have not preserved that founding heritage unchanged, but have instead transformed it through the powerful forces of genetic drift, founder effects, and the random variation in reproductive success that operate with particular intensity in small, isolated populations, concentrating some variants to high frequency, eliminating others entirely, and producing a biological profile that is in many respects unique among human populations of comparable size anywhere in the contemporary world. What the DNA reveals about the Amish is not that they were living a lie or that the traditional understanding of their origins was fabricated. It reveals something simultaneously more complicated and more interesting, that the people who crossed the Atlantic in the 18th century to build a new life in Pennsylvania were themselves the products of a much more turbulent and much more diverse history than the clean founding narrative acknowledges, and that the 3 centuries of deliberate separation from the surrounding world that followed that crossing have produced biological consequences. In disease, in longevity, in metabolic function, in the extraordinary natural experiments that their gene pool makes possible, that the medical and scientific communities are still only beginning to fully understand and appreciate. The Amish have been a mirror in which the modern world has seen reflected its own anxieties about technology, community, and the pace of change. The genetics suggests that they might also be a window into human evolutionary history, into the biology of aging and disease, and into the remarkable things that happen when a small group of people carrying a particular slice of the human genome decides to remain apart from the world and let the biology run its course across the generations.